Okay, so hello everyone. So this is the last week of the problem solving session for the course tissue engineering. I am Chandra, and briefly, I am a PhD scholar, the Department of Chemical Engineering, and I'm working in the area of tissue engineering. And this is the last week of the session, and the timing is 6 to 8. Meeting link is with you. The recording of the session will be uploaded in my YouTube channel. So please use your mic to like discuss any queries if you have, as the chat option is not available. Okay, so what we seen in week 8? Let me quickly refresh your memories. We first saw about the bone tissue engineering aspects, like what is the structure of the bone, what are the main cell types present there, the ECM component, and if we were to engineer artificial bone tissue for the purpose of repair or replacement of a critical size bone defect, then what factors should we take care of? So we discussed that. Similarly, for vascular tissue engineering, we also discuss similar things, like we discussed the anatomy of our vascular system in our body, the hierarchy, how the blood vessels are present, like from the aorta to capillary sizes. And then we also discussed individual structure of the blood vessels. What are the layers present, the resident cell types, what are the functions of these cell types, and if we had to, and what is could be the common reason for which the vascular tissue engineering like theories can be applied, like for the diseases like atherosclerosis. In those aspects we can use the principles of vascular tissue engineering to create or to create a replacement or to repair a damaged area of our blood vessels. Similarly, for corneal tissue engineering as well, we saw the structure of the eye, the corneal structure, what is the structure of the corneal epithelium, right? And we usually target the corneal epithelium to like if there is a damage in the corneal epithelium, we target that. And for that, we try to see biomaterials which mimics the properties of corneal epithelium. And in this way, we can help repairing a critical size defect in our eye so that we see better. So we will now start with some problems. So I have prepared plenty of problems for this week, which are very easy in my sense. Like if you have seen the lectures, they are very easy, but also it will help those who haven't seen the lectures yet. So it will help them, grasp the basics of the topic, and then later on in detail they can go and see the lectures and know more. All right. So let's start with the first question. Which part? So, I have divided the questions, like first few questions will be on the bone tissue engineering aspects. Next, we will look into vascular tissue engineering aspects, and then later we will go through the questions of corneal tissue engineering aspects. Okay. So now, for the first part, we are going to look into some questions that is related to the structure of the bone, as well as the biomaterial properties, cell types, and all that we should use for constructing artificial bone tissue. Okay, so first question is this: which part of the bone contains the marrow? First, periosteum, B, compact bone, C, medullary cavity, D, epiphysis. Which part of the bone contains the marrow? The bone marrow that we all know, where it is present in the bone.
I may say D.
Okay, actually the correct answer is C, right? So if you see, this is the structure of the bone, and periosteum is the outermost layer of the bone. You can see this kind of a film that is coming out. Let me quickly, give me a second. So this so periosteum is this. The membrane-like structure is the periosteum. Then this is the middle region of the bone that is called diaphysis. Epiphysis is the distal regions, all right? The proximal and the distal regions, the ends of the bone is called epiphysis, and inside that the deepmost layer is called the medullary cavity, and into it contains the yellow bone marrow. And periosteum is the outermost, then comes the endosteum, and inside the endosteum there is medullary cavity, and inside the medullary cavity there is bone marrow. All right, that's why the answer C is correct here. So moving forward to the next question. So what is the outermost layer of the bone called? Endosteum, periosteum, trabecular osteon. Yes, Sukandar, can
B,
you
ma'am.
answer? It's yes, it's B, it's periosteum. So as I showed you in the diagram, periosteum is the outermost layer and it's like a film, that's covering the entire bone. What type of bone tissue forms the dense outer layer of bones? The calcareous bone, compact bone, spongy bone, and cartilage. Now, this is the if you like differentiate the bone structure depending on the density, then you will be able to answer the layers now. There is a dense outer layer, and there is one slightly, relatively less denser layer of the bone that is seen in like cranium of our like skeleton, or in other parts as well. What it is called, or what it is formed, composed of.
Is it B, ma'am?
Is it B? Yes, it's B. It is actually called compact bone. Similarly, if you go to this structure, you will see that there is a layer called spongy bone, which is more porous. And then, if you look closely, encasing the spongy bone, there is a compact, no, more less spongy, relatively less spongy, hard, relatively more hard layer of bone that is called compact bone. Okay, so it's like compact bone first, then a layer of spongy bone, and then again a layer of compact bone. In this way, the structure of the bone is protected, like very well protected. So, what is the name of the structural unit of compact bone? What it is called? Osteon, lamellae, trabecular, lacunae.
D, lacune.
So, actually, the structural unit is called osteon. So, here I've given the structure. If you see the inside, this portion is the portion of spongy bone or cancellous bone, and outside the spongy bone is the compact bone. And one section of that compact bone I've shown, that is called osteon. All right. And the structure of osteon is actually, osteon is the site where the bone cell osteocytes reside. Okay, so inside the osteon, the osteocytes resides, and I'll come into the structure of the osteon in a later question. Okay, but you should remember the structural unit of the compact bone is called osteon. All right, these are actually like this lamellae and lacuna, these are actually part of osteon. Trabecular is actually part of spongy bone. Okay. Now, what are the small cavities in bone that contain osteocytes? So I told you osteocytes, they resides in osteon, which are the structural unit of compact bone, right? I told you this. Now, this question is based on the structure of the osteon, how the osteon looks like. So, let me go back here. So the osteon has structures like uh, canaliculi, lamellae, and as running through the center is called Hervasian canal. So it's like this, something like this. If we look from top, it's circular in nature. There is a empty canal, all right, Hervasian canal, through which the blood vessels, the veins, and the arteries flow. All right, it helps in supplying the oxygen and nutrients. And outside, there are concentric circles. Okay, concentric circles. And these are called lamellae. These circles, these rings of matrix is called lamellae. Okay, and between the spaces of lamellae, concentric rings of lamellae, there are spaces that is called lacuna or lacunae in plural. Okay, so these are the spaces, and these are the and there are also something called canaliculi. So these are some kind of fibers that exist in between the in between the adjacent lamellae, and these are called canaliculi. And where is osteocytes here? Osteocytes basically live in the lacuna. Okay, these spaces that are formed between adjacent lamellae. So these are osteocytes. Okay, this is very um, rough diagram of osteon. Later on, I'll show the structure of osteon in a better way. But now, if you are able to visualize that, how the osteon look like, and what are the structures of osteon, now can you answer where in the osteon the osteocytes reside? Is it lamellae, lacuna, Hervasian canals, or trabeculiculi? Where does it reside? It resides in lacuna, right? I just told you, isn't it? Did you understand the structure which I tried to show you with diagram? If not, so let me see if I have a better diagram here. So yeah, this is a better diagram. So if you see, this is the outer portion that is called the cement layer. Okay, and inside there are these concentric circles of matrix. These are called lamellae. Running through the center of the osteon is called the Hervasian canal, and it contains the blood vessels, lymph vessels, all right, nerves, like all these things are running through it. That supplies oxygen and nutrients, helps removing the excretory products, as well as responsible for like giving sensory stimuli, right? Then these tiny projections that are coming out of the osteocytes, these are called canaliculi. Okay, lacuna are basically the spaces in between the lamellae, and they are the space housing the cells osteocytes. So osteocytes are present at the lacunae, and from the osteocytes comes some kind of network like projections These are actually coming from the cytoplasm of the cell. These are called canaliculi. The running, the hollow region running through the osteon is called Harvesian canal, that contains the blood vessels, lymph nodes, nerves, right? The concentric circles, right, which are composed of bone matrix, which are these are called lamellae, and the outermost region of the osteon is called cement layer. Okay, so if you have understood the structure of osteon and where the osteocytes lives in the osteon, let me know if you have understood, and if there are any queries, then also you let me know. Is it good? Yes, no?
Yes, ma'am.
All right, okay. So the next question, which cells are responsible for breaking down bone tissue? So you know there are four types of cells in bone, and each cell has their own function, and every function is equally important. And so here I have listed only. So chondrocytes is a cell type of cartilage, and these are the three cell types of bone. If you can recall, which of these cell type has the function of bone resorption? Breaking down of bone can be also termed as bone resorption. So which cell type has the function of bone resorption here, if you recall from the lectures?
Osteoclast.
Yes, you're correct. So osteoclast cells, they're responsible for bone resorption, and they are found at the bone surfaces which are old and injured, and they need to be replaced, right? So they are found near those bone surfaces. They are also called osteocytes, just which reside in the osteon, right, in the lacuna region of the osteon. They are responsible for maintaining the mineral concentration of the matrix. So what is the mineral content? It is the concentration of the hydroxyapatite or the calcium phosphates, right? So these inorganic content, the content of hydroxyapatite, they are maintained by these cells, osteocytes. Okay, so and you know the location of the osteocytes as well. Then there are cells called osteoblasts. Now these cells are responsible for formation of bone, and they are present in the growing portions of the bone, like the endosteum, the periosteum. The outer periosteum is the outer membrane, endosteum is just the layer beneath the periosteum. So wherever the bone is trying to grow, from whichever region, the osteoblast will be there. And there are cells called osteogenic cells. These are stem cells kind of nature, right? They are stem, they have the stemness characteristics. They are themselves do not work. They are since native and naive in nature, undifferentiated in nature. So that is why they differentiate themselves and form the osteoblast cells. And these osteoblast cells then will continue its function of bone formation. All right. So predominantly these three cell types work, they are differentiated, all right, and they have individual functions to maintain this homeostatic condition of the bone. But osteogenic cells, they are only activated when they are required. Okay, that is why you see the location, they are located in the deep layers, like they have their own niches in within the bone, which protects it, all right, from any other environmental cues, and they're activated only when needed. Now, if you were to design a scaffold for bone tissue engineering, then which of these properties should it possess? Non-toxicity, induced vascularization, elasticity, mechanically strong, porous, and should degrade faster. Which of these properties are important for a scaffold to be used for bone tissue engineering? There are more than one option that can be correct here. Yes. Should it be non-toxic? Yes or no?
Yes, ma'am.
Yes. What else? What else should it possess? There are more than one option that is correct.
I would prefer A to E, but I'm not sure about the F last option.
Okay. Actually, all of these options are correct here. When we're talking about a scaffold, a tissue engineering scaffold, actually tissue engineering construct, it if it shows a degradation property, then it's good. Means as it degrades, the cells, resident cells of the tissue, the host cells, they will start secreting their own ECM. So over a period of time, you will see that the material is gone and the cells have secreted their own ECM. So what it helps, it helps preventing a secondary surgery to remove it afterwards. Whereas if you use stainless steel rods or something like that for repairing the bone defect, a fractured bone, then a secondary surgery is required to remove stainless steel, these kind of alloys, because they do not degrade at as much fast. So ideal thing is the degradation rate should match the rate of bone formation, right? Otherwise, if it degrades too fast, by that and within that time, if the new collagen and hydroxyapatite is not secreted enough in enough quantity, then the newly repaired bone will be mechanically weak and will be susceptible for future damages. Similarly, if it degrades very slowly, then what will happen? It will act as a foreign body, right? And over a long period of time, it can show some local and systemic immune responses. So, and it will be, and so in that cases, it will like cause the surgeons, like it will im- um, it will force the surgeons to do another surgery to remove that implant. So that's very painful for the patient. So this degradation property is also one of the properties, or one of the ideal properties for a scaffold to possess in order to be used in bone tissue engineering, right? Oh, sorry, elasticity should not be there. Elasticity we don't need. I forgot to mention. Why elasticity should not be one of the properties? Because bone is a rigid material, right? There is no need for bone to bend. So elasticity should not be one of the properties. Except elasticity, all of these properties are important for a scaffold to possess to be used in bone tissue engineering. All right, I just got that cleared first. Any queries here? Do you have any queries?
Ma'am, what could be the pore size that can mimic the extracellular matrix? The range.
So should be greater than 100 micron.
Okay.
All right. Yeah. Anything else? All right. This is the next question. Which of the following is a mechanism by which cells added to bone scaffold can enhance bone regeneration? A. Secrete osteogenic and vasculogenic molecules and growth factors. B. Induce inflammatory response. C. Induce bone resorption. D. All of the above. So we are talking about the mechanism by which cells can actually help in bone regeneration. So which mechanism or by which mechanism cells are helpful? Only one is correct here. Or rather, think in this way, why would you consider adding cells along with the biomaterial scaffold? How would it help?
Option
Option A, yes, you're correct. We want the cells to secrete the essential osteogenic and vasculogenic molecules so that the process of osteogenesis can happen, like the process of new bone formation can happen, and vasculogenic molecules can help in growing new blood vessels, right? And what is not required from the cells, that it should not induce inflammatory response. So we can use immune privileged cell types for this application, or we can use autologous cells, all right, to prevent inflammatory response. And also the cells, if we add along with the scaffold, it should not immediately stimulate bone degradation, right, or bone resorption. The bone resorption should be done by the host cells. The host cells, they should degrade the bone as the new bone formation is happening. Like the cells that you're adding from outside, they should not do that. Means why? Otherwise the mechanical structure and all, mechanical properties, they will get compromised immediately after implantation, so it is not required. Okay, this is the next question. Research shows that injecting chondrocyte to bone defect inhibit vascularization. Is it true or false? So you have a bone. Yes, yes. Is it true or false? Can it be? Actually, it's true. All right. So there is a research work that was discussed in the lecture by Joseph Vacanti's group, and they tried to inject both differentiated osteoblast cells and differentiated chondrocyte cells at the site of defect. Like directly, they did that, and what they observed is the site where chondrocytes were injected, there the new bone formation was not as good as that, compared to that site where osteoblasts were injected. Why? Because it didn't um, show, the site didn't show any vascularization. It rather inhibited vascularization. Okay. So if you know chondrocytes, they are the resident cells of cartilage, and cartilage itself is avascular tissue. The chondrocytes, they are not habituated living near endothelial cells. So that is why, I think that is why they didn't promote vascularization. But the idea that why did they think of injecting chondrocytes as well as, like, along with the bone cells, means as as a test parameter, is because bone can form along the osteoblast pathway or along the chondrocyte pathway as well. So you know there are cartilages, right, that encases the ends of the bone. And as we grow older, so when we are young, that cartilage is very weak, very naive state. As we are growing, that cartilage, it starts forming a cartilage plate. And as the plate is formed, it means your bone cannot grow any longer. So that's the one pathway through which bone growth can happen. All right. And in adults, a distant cartilage plate is present. So they thought maybe that pathway, through that pathway, using cartilages or using chondrocytes, the bone defect can get healed. But it was not so. It was not as prominent when they directly used osteoblast. So this research laid a foundation on what type of cells would be good to be used for tissue engineering. So you will see nowadays most of the people are using osteoblast, all right, and they are also using mesenchymal stem cells because they're osteogenic cells, means they are undifferentiated cells, and once they are in correct environment, in a correct microenvironment, they can actually proliferate more and more and differentiate into more osteoblast cells. So the purpose uh, of bone formation can be achieved when you deliver osteoblast cells. All right. So that is like very well proven, and repetitively it is being shown with biomaterials of different origin in different animal models. You will see like it's a like known concept, all right, in bone tissue engineering. All right. This is the next question. Blank involves recruitment of MSCs for differentiation into precursor cells, and then blank. So first blank will be filled by the process name. What process helps in recruitment of MSCs? Then these MSCs give rise to a form of differentiated cell. And what is that differentiated cell called? Okay. A. Osteogenesis, comma osteoclast. B. Osteoinduction, comma osteoblast. C. Osteoinduction, comma osteoclast. D. Osteoconduction, comma osteoblast. Which of these options will fit correct and make sense of this statement? Yes, give it a try.
Is it option B, ma'am? That given
Option.
osteoblast.
Okay, so let me see. Osteoblast. Mm, let's see. Okay, I think it is B. Yeah, it's B. So MSCs will definitely differentiate into osteoblast, and the process through which this recruitment of MSCs starts is through osteoinduction. So in order to understand why osteoinduction is correct. So osteoblast, I think it is self-explanatory. I've already told you that MSCs, they can differentiate into osteoblasts. That's like proven through experiments. But what do you mean by these terms like osteoinduction, osteoconduction, osteogenesis? Like what do these terms mean in the context of bone tissue engineering? So to explain that, I've used these diagrams for this slide. Osteoconduction means the material that you are using, it should have the ability to act as a skeleton, act as a framework. Okay, so it should give the cells a template onto which they can grow. So if you were to remember what do you mean by osteoconduction, remember the terms like template, framework. So it will help you understand what this osteoconduction means. Now, when the scaffold has given a framework, then it should and given a framework to the cells. Now, what the cells will do? Now, the cells have come here, the scaffold, they should induce new bone formation. Okay, so that is the next step, osteoinduction. The recruited cells should be stimulated to secrete growth factors and other signaling molecules so that the process of osteogenesis or new bone formation can happen, all right, over the scaffold. So that is osteoinduction. And osteogenesis is then the next step. That is when the ECM of the bone and more bone cells that gets formed, that is the osteogenesis. So if we see sequentially, you transplanted a scaffold. First, it should osteoconduct for for the host cells to migrate. Then the host cells should induce the secretion of, the host cells should get induced to secrete growth factors. All right. And these host cells will get stimulated inside the scaffold. That is called osteoinduction. And once the induction process is complete and the cells are activated with by secreting many more growth factors, what it will do, it will recruit more cells. If maybe of stem cell origin, these stem cells, they will proliferate and uh, give rise to more differentiated osteoblast cells and other osteocyte cells. So these cells will now help in secretion of bone matrix like collagen and hydroxyapatite, as well as more osteoblast cells will be there, and they will also help in secretion of or formation of bone. That is last step is osteogenesis. So your scaffold, for successfully healing the defect, your scaffold should have all these three properties. All right. So osteoconduction, it should support the cell growth. All right. Or it should allow cell adhesion and subsequent cell growth. Osteoinduction means and it should allow stem cells, if the stem cells are there in the host, or it should allow the stem cells that you supplied with the scaffold, they should get differentiated into osteoblast cells and secrete more and more growth factors to recruit more osteoblasts. That is osteoinduction. And osteogenesis means the secretion of bone or formation of bone. That will be done by the osteoblast cells and other resident cell types. Okay, it's osteogenesis. So the first step, that osteoinduction, that actually helps, not the osteoconduction or osteogenesis that will come here. It is osteoinduction that will be appropriate here for this question, because during osteoinduction, the MSCs, they get stimulated, they get recruited. All right. And once they are recruited, they will start differentiating in presence of some growth factors. So yeah. All right. Is it clear? These are very interrelated terms. As you understood, very fine lines exist, or fine, um, what do you say, boundaries exist between these three terms. If you could understand the result that each of these term is indicating, then it should be fine. If there are any queries, let me know in understanding what is osteoconduction means, osteoinduction means, or osteogenesis means. Any queries? Okay. I'll assume that you have understood. If not, feel free to ask any questions. All right. This is the next question. Which of the following is a characteristic feature of osteoconductive materials? Right. Again, this question is based on whatever you have understood in the previous slide. They stimulate new bone formation. They serve as a template for bone growth. They attract and differentiate osteoblasts. They release growth factors to promote bone healing. Which of these option is correct, or correctly fits the definition of osteoconductive materials?
B.
Yes, B. Yes, you are correct. Osteoconductive materials means it should act as a template for bone growth. And then the subsequent processes like osteoinduction, osteogenesis will happen. Then this is the next question. What is the advantage of using mesenchymal stem cells in bone tissue engineering? Why should one prefer to use the mesenchymal stem cells? Like what they do, they first make the scaffold, then add mesenchymal stem cells, allow it to grow on the scaffold first, and then transplant the entire thing. So what's the advantage in doing that? A, their ability to differentiate into osteoblast, their immune privilege status, their ability to secrete growth factors, all of the above. Which of the option do you think is correct and matches um, with what is asked here? Yes, go ahead, give it a try. Why would one choose mesenchymal stem cells? Okay, if you're not getting, let me answer it for you. So it is because of all of these reasons. Mesenchymal stem cells, they have the ability to differentiate into osteoblasts like very well. You can do it in vitro, in vivo. The result will be same. Then they are immune privileged, means they do not contain the IHC complexes through which the body's immune system cells can recognize them. So they can hide, or they can hide themselves very well from the body's immune system and will not elicit immune rejection quickly. All right. Then, once stimulated, they will secrete growth factors. So, that is, they will provide the tissue engineered scaffold the property of osteoinduction as well. That is why, if you use mesenchymal stem cells along with the biomaterial scaffold, then you will gain, or you will faster your response, wound healing response, or defect repairing response by integrating all of these responses. Right? Right. Is it clear? Other questions are like, is the topic was becoming clear to you all? If there is any
Yes, ma'am.
query, yeah, okay, all right. As I was saying, if there any query, let me know. All right. Which of the following are the growth factors used in bone tissue engineering? So sometimes only the biomaterial and the cell may not be sufficient for the application. You may need to additionally add some growth factors within your scaffold that may help in your application, like maybe faster your wound healing process or something like that. Like that is why this is the triad, right? Biomaterial scaffold, cells, and factors. They all together are the triad or the ingredients for making an artificial tissue. So, which of these growth factors are generally used or most widely used in bone tissue engineering? EGF, epidermal growth factor; NGF, nerve growth factor; C, bone morphogenetic protein; D, vascular endothelial growth factor. Which of these is most widely used? I think here uh, more than one is correct here. Okay, I forgot to write. Then one correct. Okay, one or more options are correct here.
Is it C and D, ma'am?
Yes, you're correct. So C and D are the growth factors which can help in regeneration of the bone faster. So C is BMP, and BMP is uh, the growth factor responsible for differentiation and migration of osteoblast. So it can trigger the naive osteogenic cells to get differentiated and migrate and form the osteoblast cells. So BMPs exist in BMP2, BMP7. So these are the different variants of BMP. Then VGF, that is vascular endothelial growth factor. So that is a factor responsible for uh, enhancing the functions of endothelial cells. So it can cause the endothelial cells to migrate, proliferate, and function better within your scaffold. So that is why these two are used when our goal is to promote osteogenesis as well as vascularization through our scaffold. So we can use these two factors. Apart from these two factors, there are other growth factors that are considered. So that is HGF, hepatocyte growth factor, IGF-1, insulin growth factor 1, PDGF, platelet derived growth factor, is AB and BB, or these are the variants. Then TGF beta as well. So these are some of the other growth factors that one can consider, and given here are the functions of these growth factors as well. All right. Then BMPs, they are primarily responsible for what? A, angiogenesis, B, osteogenesis, C, neurogenesis, D, chondrogenesis. For which of the options? BMPs help in which of the processes? Actually, we are running out of time, and we have more questions. So if you can, don't hesitate. Just these are very simple questions and um, very basic questions from for this week's lectures. Whatever you're
Ye-
able to understand. Yes, is B.
yeah.
Osteogenesis. It helps in osteogenesis. All right. Means formation of newborn. This is the next question. Which of the following commercial product uses BMPs as growth factors for bone regeneration? So these are the names of some commercial products uh, which are used in clinical settings, and a few of them here also more than one is correct. So one or more of these commercial product, they uses BMP as one of the constituent. Okay. Actually, the correct answer is this OP1 putty. So if you go back to the lecture, you will see that it is discussed thoroughly there. But in brief, this Nova Bone, this product, it is mainly composed of bioactive glass. HTR, or heart tissue replacement, it is mainly composed of PMMA, polymethyl methacrylate. Then alpha BSM, it is composed of calcium phosphate cement. All right, so it is a ceramic, it is also a ceramic, whereas PMMA is a polymer. Whereas OP1 putty, it consists of the organic collagen component and BMP7 component. So these two together are used in OP1 putty. So this is the product that uses BMP as a growth factor. All right. Now it's the next question. So we have now looked into all the questions for bone tissue engineering. Now we will move to some of the questions related to vascular tissue engineering. All right. First question is this. Very simple. What is the primary goal of vascular tissue engineering? Why do you need to use vascular tissue engineering principles? First is to regenerate muscle tissue, to generate or replace damaged blood vessels, to promote nerve regeneration, to promote creation of bone scaffolds. Why do you need vascular tissue engineering? A, B, C, D. Which one is correct?
B.
Yes. Very easy. You need vascular tissue engineering principles to restore or repair or replace any damaged blood vessels in your body. Correct. Which of the following is correct combination of vascular cell type and its location? So this question is talking about the structure of a blood vessel. So there are layers. These layers are called tunica intima, tunica media, and tunica adventitia. I've named them from the um, um, tunica intima is the layer which is directly in contact with blood, the flowing blood, whereas tunica adventitia is the farthermost layer, the outermost layer. Okay. Now each of these layers they have a primary cell type present in it. So you have to match it basically, and and then after you match it, you can correctly answer this question. A. Endothelial cells present in tunica adventitia. Is it? You have to answer. Then fibroblast cells is it in tunica intima? Then smooth muscle cells is it present in tunica media? And D. None of the above. None of the above are a correct combination, a correct matching. So I told you. So there are three layers of a blood vessel. So I drew it in a planar form, but actually it's like this, right? And here is blood. This layer, the layer which is directly in contact with blood is called tunica adventitia. Sorry, is called tunica intima. Sorry, is called tunica intima. Intima. Then the outermost layer, just farthest away from the blood, is called tunica adventitia. And the middle layer is called tunica media. And each of these layers have specific cell type. I'll tell you later, but if you can guess which is correct here, then we'll go forward. Yes, go ahead, give it a try. Which of this is correct here? Okay, don't worry, I'll answer it for you. The correct answer is option C. The smooth muscle cells, they are present in tunica media. All right. Yes, smooth muscle cells are present in tunica media, the middle layer. Intima layer is the layer which is contact in blood. So it is that's why lined with endothelial cells, and the outermost layer composed of fibroblast cells. All right. So I think we got it cleared now. Now, why arteries have a thicker layers of smooth muscle cells? So you know the hierarchy. So largest uh, blood vessel in our body is aorta. From aorta, there are arteries and then arterioles and then capillaries. These capillaries meet and collect all the waste, deoxygenated blood, and these deoxygenated blood then goes to venules and then um, ventricles and then veins, and then it pumps back to our heart to get oxygenated again. So that's the hierarchy. Now arteries, for them, the adventitia layer is relatively thicker when you compare it with arterioles and capillaries. Capillaries are generally a monolayer of endothelial cells. Then, as you go to arterioles, the smooth muscle cells form the media layer, but there are only a few layers. Whereas when you look at the structure of arteries and arterioles, which are very thick in nature, like the diameter is very thick, close to, I guess, 2 mm, 3 mm. So for them, the thickest layer is the adventitia layer, and there are almost 5 to 10 layers of smooth muscle cells are there. But why is it? Why? What is the reason for that? If you can guess. Option A, to withstand high blood pressures. B, to encourage more nutrient diffusion. C, to become more thicker in diameter. And D, none of the above. Why do you think could be the reason that arteries or aorta they have thicker layer of smooth muscle cells?
A.
A, yes, correct. So smooth muscle cells, they help in contraction and dilation, like relaxation, right? Uh, and as the blood is pumped out of heart, it rushes through very quickly. So the blood pressure at arteries or arterioles, they're very high, close to 120 mg, millimeter of mercury. mmHg is called. So it is almost close to 120. And as the blood is flowing through different hierarchies of blood vessels and reaches capillaries, there the blood vessel, there the blood flow at a very low pressure, like close to 20. All right. So that is why the capillaries that even they have a monolayer of endothelial cells, they can still sustain the flow of the blood without getting ruptured. And since they are monolayer of endothelial cells, so nutrients can easily diffuse out and oxygens can easily diffuse out to the neighboring or surrounding tissues. But as we are going up, like towards the structure of aorta, there the to withstand that variation in blood vessel blood pressure, means how fast and the blood is flowing, and to avoid rupturing as well as that, the smooth muscle cells helps in giving that ability to contract and dilate accordingly, so that elasticity to the blood vessels is needed to withstand this high amount of blood pressure. So that is why um, the arteries and the aortas, they have very thick layer of tunica media. All right. This is the next question. What is the role of smooth muscle cells? Why smooth muscle cells are important? A, to form the outer layer and provide contractile function. B, to promote blood clot formation. C, to form the endothelial layer. D, to degrade the scaffold. Why would smooth muscle cells be required in the structure of the blood vessel? It's very easy. Come on. We just discussed. So I'm not able to understand. Is the questions too easy that you don't want to answer? Is like, oh, it's not up to my standards, or the question is too difficult that you're not able to understand at all? What's the issue here for this unresponsive nature? It's fine. You can be frank. It's totally fine. What's the role of smooth muscle cells? Go ahead. Yes. Correct option is option A, right? Now, this is the next question. What do you mean by the disease that is termed as arteriosclerosis? Okay, what happens in arteriosclerosis? It is the formation of plaque that can partially or completely block the lumen of arteries. B, it's the formation of plaque that enhances endothelial cell activity. C, formation of plaque that modifies the structure of blood vessels. D, the formation of plaque that enhances the immune cell function. So which option correctly defines the condition that is called arteriosclerosis?
A.
A, it's actually A. Okay. So here what happens, if you see like this, if this is the intima layer, and here they are the blood endothelial cells over in people who eat too much fat, or who are maybe genetically susceptible, for them the secretion of cholesterol is very high. All right, and that is one of the reason why atherosclerosis happen, by the way. So there are two types of cholesterol molecules. There is low density lipoproteins and high density lipoproteins. So these are good. Okay, these are good for your body. But whereas low density ones, they are not so good. They are basically, if this content is quite high, then then the person is more susceptible for this condition, atherosclerosis. So this cholesterol molecule LDL, they binds with like macrophage cells and other type of ECM protein, and they start accumulating. Okay, over a period of time, it starts accumulating in one of the surfaces, in it like with on the surface of the endothelial cells. And what happens, the blood flow that is going to now take place through this lumen is going to be compromised. Like, it's not, the blood pressure is not going to be same as the normal one. The blood vessel itself will have a very hard time to contract and relax. So because of which you can have problems like heart attack or cardiac arrest. So these things can happen because of it. So there are various treatment options are available, but for tissue engineering as well, regeneration engineering as well, you can target this problem of atherosclerosis, right? So these are the this question actually talks about the treatment options which are available for atherosclerosis. So, what are the different strategies that people use conventionally to treat this condition? A, angioplasty; B, stents; C, arterial reconstruction; D, vasodilators; E, anti-proliferative agents. Select all that apply, means more than one option is correct here.
B.
Yes. So, most commonly, actually, all of these are correct, but let me discuss one by one. So, since you said stents, we'll start with that. Stents are basically a cage, a metal cage, usually made up of some biodegradable metals, and they are deployed through through a guide wire connected to a balloon. And once it reaches the site of the plaque, the surgeon inflates the balloon, and the stent basically expands and holds the blood vessel. All right. uh, like, it basically, what it does, along with the plaque, it mechanically causes the blood vessels to open up so that the normal blood flow pattern can be restored. All right. So that is why stents are very normally used for atherosclerosis. There are also nowadays drug-eluting stents. So what happens at the inner surface of the stents, they are coated with some polymer, and this polymer may be having some concentration of drug mixed with it. So as the blood is flowing and causing some shear forces on the surface of the stent, this drug starts coming out. and these drugs may be used to target the cholesterol molecules and the other content of the plaque, so that over a period of time that plaque gets dissolved, okay, and removed with the blood, with the flow of the blood. So this is an advanced version of stent, that is drug eluting stents. Apart from stent, people also do angioplasty, where they like basically, angioplasty is the procedure where people through this people can deploy the stent, or what they can do, they can directly inject the drug at the site of the atherosclerosis where the plaque is formed. All right, so through this procedure as well we can dissolve out the plaque. Then if the plaque has built up so much, like it's completely blocked the lumen of the blood vessel, then the surgeon need to remove that entire blood vessel portion. Right, they first image it using angiography. So they first find the location of the plaque where it is, and then surgically remove that, and reconstruct that removed portion by taking body's own blood vessel, all right, from anywhere in the body where it is safe. So they take it and reconstruct it, that region. So it's like we are replacing the entire area with a uh, better blood vessel lumen, right, from the body. Then people also use drugs like vasodilators. So these drugs basically act on the smooth muscle cells, right, modulate those function of smooth muscle cells and causes it to dilate. All right, and what happens due to that, the diameter of the blood vessel increases, and through that the blood flow can be restored to normal. And there are also drugs that is called anti-proliferative drugs. So they basically prevent the further uh, growth of plaque. So within the plaque, I told you there are some cells as well, the immune cells and like macrophages, etc., who are basically proliferating in order to. So why would macrophage come at the site? Because they may think that it's maybe they get adhered to it. I don't know the exact reason why macrophages will come at the site of the plaque, but I think maybe because they get adhered to that surface, maybe it's more sticky, or they get sensed or they get alarmed that it's a threat. So they try to maybe kill it on its own, like by phagocytosis. So in order to prevent further proliferations, as the cells will do its function, they will proliferate, right? So in order to prevent that, they use and these proliferative agents, so anti-proliferative agents, so to prevent further buildup of the plaque, basically. So all of these options are correct here, and these are some of the strategies that are currently being used to treat atherosclerosis. Now, in vascular tissue engineering, which mechanical property is crucial to ensure proper function of engineered blood vessels? First, electrical conductivity, B, elasticity, C, thermal conductivity, D, optical transparency. What mechanical property actually is crucial for any scaffold and tissue engineered construct that you have made, that you are going to use for engineering blood vessels?
B.
Yes, you're correct. Yes, you're correct. So we want the biomaterial to have this elasticity property, and this elasticity property should match with the native blood vessels, so that it can contract and dilate accordingly to the flow of the blood. Okay. So whereas thermal conductivity, electrical conductivity, all these things are not an essential property when you're talking about engineering blood vessels. Then keratocytes. So now we have completed the vascular tissue engineering part. We have now going to start the corneal tissue engineering part, and we are going to see some problems based on that. So keratocytes, they are basically the resident cells of the stroma region of the corneal epithelium. All right, I'll talk about the structure of the cornea in a minute. So keratocytes, they are the cells that help in ECM regeneration during stromal damage. Right? Is the statement true or false? Like try, it's only 50% chance um that it's good. Just give it a try. Even if you don't know the correct answer, is fine. Just give it a try then.
True.
Yes. So you can be only 50% wrong here. So it's actually correct. Keratocytes, they do help in ECM regeneration. All right. What are the what is the first stage of corneal wound healing? So corneal wound healing is just like the normal wound healing process we already know. So what will be the first step for that? A, hemostasis; B, cell proliferation; C, inflammation; D, remodeling. What's the first step of
And
wound healing? A. Yes, you're correct. It's hemostasis. Right? The first stage of wound healing is hemostasis. Now, like I have made some questions based on the structure of the cornea. All right. So, following questions will be based on that. Which of the following is the outermost layer of the cornea? So, these are the options: Descendants membrane, Bowman's layer, stroma, epithelium. Which is the outermost layer? I'll show the diagram and all in a minute, but give it a try first.
D, ma'am.
D. So, why is it? Let me show you. So this is the diagram that shows the layering of of the corneal region. So, eye has the cornea, that is the most essential part through which light can uh, come through and we can see. Right behind the cornea, there is iris and pupil, and there is lens. So, what like how the structure of the cornea is made? So, outermost layer is the corneal epithelium, which is directly in contact with tear film. So it's protecting the eye from any from the invasion of pathogens or dust particles, basically protecting the eye from any damage. Then below the epithelium, so epithelium has some cells types, like epithelial cell types are there. Below it, there is called Bowman's layer. So Bowman's layer is basically ECM, uh, only ECM layer. There are no cells here. Okay. Then below Bowman's layer, there is stromal layer, a stroma, epithelium stroma. So it is the thickest layer of the cornea, and which is why it will impart the corneal tissue, mechanical strength. The resident cell types is keratocytes. Okay. And just beneath stroma, there is a layer called Descement's membrane. Again, like Bowman's membrane, this layer is only ECM. There is no cells here. And just below the Descement's membrane is the endothelium, that is lined by endothelial cells. So it is the thinnest layer. All right, thin layer of endothelial cells, and it is basically squamous. Squamous, like flattened like an egg omelette, like that. So it's squamous endothelial layer, endothelial cell layer is there, which is very thin. So that is the endothelium layer. So if we go again, outermost layer is the epithelium, then there is Bowman's layer, then there is stromal layer. The stromal layer, there is keratinocytes. Beneath stromal layer, there is Descement's layer, and beneath it is the endothelium. All right, makes sense? Moving forward, the stroma or the corneal stroma makes up approximately what percentage of the cornea's total thickness?
C.
C, yes, you're correct. So the thickest portion of the cornea is stroma. So whichever option seems highest to you, that's correct. Actually, it's in the lectures they said around 70%, but yeah, 70-80% is fine. That percentage is fine. Then what type of cells make up the corneal endothelium layer? Okay, we're talking about the bottommost layer of the corneal tissue. A. Stratified squamous epithelial cells. B. Simple squamous endothelial cells. C. Columnar epithelial cells. D. Cuboidal epithelial cells.
B, ma'am.
B. Yes, you're correct. Is the endothelial cells, which are basically simple squamous. Right. Towards the primary function of corneal epithelium, to maintain corneal hydration, to act as a protective barrier against pathogens and injury, to provide structural support, to facilitate nutrient absorption.
B, ma'am.
Yes, sir, correct. Right? It is going to act as a barrier against pathogens and injury. Which factor does not contribute to cornea's ability to refract light? So, light reflection is very important property of the cornea that helps us see. But which of this property is not essential for that light refraction ability? A, its curvature. B, its transparency. C, the presence of blood vessels. D, its smooth surface. Which factor does not
C, ma'am.
contribute? C. Yes. It's C. So you're fast. Okay. So actually it's C. The corneal tissue um basically is avascular, at least in the epithelium region. And vascularity comes very later. As you can see, it's at the bottommost layer. So it's the curvature and the transparency and the smoothness of the surface that helps the light to refract, and we are able to see better. So defect comes in those things only, like when the curvature changed due to a condition, or when the cornea is not as transparent. That is the condition of cataract. Then if this surface is not smooth anymore, then also light will not reflect well. Then you need intervention. But since the corneal layer, the topmost layer, epithelium, does not have blood vessels, so it will not contribute anything regarding light reflection, right? Light refraction, okay. What is the approximate thickness of human cornea in the center? In the center, talking about, so if this is the eye, this is the central region. So I didn't draw a very good sphere, but I guess you get the idea. So this is the central region I'm talking about. What's the approximate thickness of cornea there?
B, ma'am.
Yes, it's around 550 micron. So from the lectures, I have taken this slide. Let me show you. So this is the slide from the lecture that discusses about the dimensions of eye, basically. So if you see the anterior, this is the anterior portion, and this is the posterior portion, and the cornea, they are trying to show the entire diameter in the anterior is around 11.7 mm, and in the posterior it is around 10.6 mm. Okay, so anterior it's kind of elliptical, note that, and when you see posteriorly it is circular. So the shape, depending on how you are looking at it, the corneal shape is different also. Then the radius of curvature. So this is the cornea, right? This is the this portion is the cornea here. So if you zoom in, this is the zoomed in version. This portion is zoomed in here in C. So here you will see that the centrally the corneal thickness is around 5.2, or sorry, it is around 0.25 mm or 520 micron around, and peripherally, like towards the edges, the corneal thickness is around 0.67 mm, that is 670 micron. The region where the wound healing studies and the defect is most widely studied is this central region, and this region is around 4 mm. So here what all the surgical intervention and all may be required, because this is the most susceptible region for any defect or condition. All right. This is the next question. Superficial cells of the corneal epithelium have which of the following functions? So we will come into the structure of the corneal epithelium only. So corneal epithelium is stratified. Note, it is stratified, just like epidermis of the skin. Like epidermis of the skin is stratified and has various layers of cells. Similarly, corneal epithelium is also stratified, and after a few days, the topmost layer of the cell can shed off. All right, and the bottommost layer again seems like to have some naive cell types that keeps on dividing and provide more and more cells to replenish the lost cells of the top layer. That is the stratification structure is there of corneal epithelium. So the topmost layer is actually called, the topmost layer of corneal epithelium has these superficial cells. But what is the function of it? A, to secrete mucus, B, to protection and shedding, C, absorption of nutrients, D, collagen production.
B, ma'am.
Yes, you're correct. So mucus is actually produced by the tear film, that is the outermost layer of the eye, covering the corneal epithelium. And absorption of nutrients, it happens through that tear film, like in contact with air and all. Um, Cells can absorb some oxygen, and collagen production is not the case for this at all. It is not at all appropriate here. So it is so this is the diagram showing the cross-sectional view of the corneal epithelium that shows the different cell layers. So as I talked about, the bottommost layer consists of some naive cells that have the capability to proliferate and give rise to large number of daughter cells. So these are the basal layers. Just above the basal layers, there is a layer of cell that is called wing cells, and the topmost layer is called superficial cells. And if you see the topmost layer of superficial cell, these cells, they have some structures which are called glycocalyx and microvilli. So these glycocalyx and microvilli structures, they are directly intertwined with the tear film. Okay, so tear film is a very, very thin layer that helps in hydrating the eye, basically. And the superficial cells, they have, because of this microvilli structure and glycocalyx layer, they get um, they tightly are in contact with the tear film. Okay. Hmm. So what is the approximate turnover rate of corneal epithelium? So how frequently do we get a new layer of superficial cells and as the topmost layer of the epithelium? How frequently does that happen? In 1 2 days, 7 to 10 days, in 1 month, or in 6 months? Give it a guess. How frequently do you think the eyes replenishes the cells, basically? It's faster than skin. Let me tell you that. If you know the turnover rate of skin, epithelial layer, or the skin epidermis is faster than that.
I think it's B.
Yes, actually, you're correct. It takes only 7 to 10 days for the new layer of superficial cells to come, right? The old one sheds off after in 7 days, and after 7 days, a new layer comes. That's the turnover rate. Corneal epithelium absorbs nutrients and oxygens from, like from where it absorbs nutrients and oxygen. The blood vessels in the cornea, aqueous humor, tear film, conjunctiva.
A, ma'am.
So actually the correct answer is C, the tear film. So if you note the structure of the not this one. Yes, this is the structure of the corneal tissue. And you, as I told you, the bottommost layer is the is the endothelium. You see how far the endothelium is from the epithelial layer. So epithelium and between this Bowman's layer keratinocytes, there is no network of blood vessels. Blood vessels start here. So it's very difficult for the epithelial layer to gain any sort of nutrition from the endothelium. So that is why it uses the tear film, you know, the structure of the tear film, which is the outermost structure of the eye. Okay. And this is the tear film. This tear film is directly in contact with the superficial cells through microvilli and glycocalyx layer. So through this tear film, the air from the air, the oxygen and the nutrients, they can diffuse, all right, into the eye, and whatever the nutrient requirements, that gets fulfilled. Okay. So it's through the tear film the corneal epithelium can absorb nutrients and oxygen from.
Okay.
Okay. So this is the next question. Which area of the eye is responsible for supplying new cells to the corneal epithelium during regeneration? Corneal, central cornea, I mean that 4 mm region. Okay. Then stroma, limbus, endothelium. From where um, new cells means must be a source in the eye where there is some stem cells that proliferate into, say, progenitor cells or precursor cells, and they further proliferate and give rise to new cells like basal cells and all, that helps in the um, giving rise to new cells and maintain the stratified layer of the endothelium, sorry, epithelium, corneal epithelium. So from where these new cells are coming in the eye, in which region of the eye?
I'm not sure, but is it A, ma'am?
So actually, the correct answer is C. It's from the limbus. So where is limbus? So you see, this is the not this diagram. The limbus. Let me show you through a different diagram. Okay, this may help. So this is the corneal portion. This one. This is the corneal portion, and just beside it is the limbus, all right, and then conjunctiva and all, and then comes the retina. Okay, so some portion will be conjunctiva, and then the remaining portion will be part of retina onto which the image is formed. So this is the limbus portion, just adjacent to the corneal region. Okay, now you know where is limbus. Now let's go back to that question. So in the limbus region, there are some stem cells that is called limbal stem cells. So although the best, so and this is the cornea and this is the they have not shown the complete layers of the cornea here. They have only showed in the diagram the layer of corneal epithelium. These are the layers of corneal epithelium. All right. So these are the limbus stem cells. They migrate, they first proliferate, and the daughter cells, as a result, they migrate towards cornea like this. Okay, over the Bowman's membrane, and then these knife cells, they become part of basal cells, basal cell layer, and then further they help in replenishing the topmost layer of the epithelium, that is superficial cell layer. All right, so the origin of the basal cell is actually from this limbus region, from the limbus stem cells. All right, is it correct? Is it all right? Did you understand from where actually the origin of new cells come from, in our eye? It comes from the limbus
Yes, ma'am.
region. What are the essential properties of scaffold for corneal tissue engineering? Select all that apply, means more than one is correct. Protection, transparency, refraction, and vascularization. If you were to design a scaffold to say replace the corneal epithelium only because that is affected, all right, let's suppose that, or if the damage is too deep, like it can also affect the Bowman's membrane and further, so then we have a stroma as well. So then we have to think like the thickness of the scaffold and on. But what are the crucial properties that a scaffold should have in order to become part of your cornea? Like you have to first think of the properties of the cornea that helps you see, and you have to mimic those properties into a scaffold so that it can act as a replacement or act as a conduit through which repair process will happen.
A, B, C.
Okay. A, B, C. What about vascularization? Do you think it should promote vascularization, or are you
Yeah,
confused?
I think it should also.
Actually, like vascularization, when we are talking about only the cornea, in the cornea you do not see blood vessels penetrating the corneal tissue, as I showed you. Blood vessels come afterwards, all right, as we go inside of the ear. But yeah, in the eye, it's inside of the eye, but eye does have blood vessels. Otherwise, how it will, function, the and how the nerves are there, how they will get nutrients. So blood vessels are there, but the corneal region, in order to maintain that transparency, the blood vessels do not penetrate. Okay, so that you see as clear as possible, at least towards the lens, the region consisting of lens. So you saw that 4 mm region, right? So just behind it, there is the optical lens through which light goes into our eye. So that region should be completely transparent. So that's why vascularization is not an important property. So if you were to replace that particular region with some material, then even if that material does not induce vascularization, it's fine, you can still use it, but it should promote protection, like it should protect the eye, the inner, so that the inner portions of the eye, the retina, the fluid regions and all, so those get protected very well. Then it should be transparent in nature, light can smoothly pass through it, and it should refract the light as well. Okay, so these three properties are essential, right, for a tissue engineered corneal tissue engineering scaffold. Now, which of the biomaterial cannot be used or is not the biomaterial for constructing a scaffold for this corneal tissue engineering? Which biomaterial is not suitable for this application basically? Collagen, dacron, hyaluronic acid, sulfibron.
See, ma'am.
Okay. So it's a good attempt, it's fine, but actually the correct answer is dacron. So dacron is a commercial name of the polymer PET, right? So this polymer is mainly used for vascular tissue engineering because it has a very good elasticity property, and you can make very fine fibers out of it and use those fibers to create blood vessels like conduits. Whereas collagen, hyaluronic acid, sulfibron, these are all naturally derived polymers, and they are very transparent in nature. Like if you have ever worked with these polymers, you will know that you can cast very transparent like hydrogels out of it. And also they are slightly like hydrogels, no, so they can absorb some water, so they can interact very well with the tear film and help in nutrient exchange and all. Okay, so collagen, hyaluronic acid, sulfibron are some of the biomaterials that are used for corneal tissue engineering, whereas dacron is not one of them, right? This is the last question for today's session. Which of the following commercial corneal replacement product uses PMMA? So PMMA is a polymer, polymethyl methacrylate. So it's again, it's a synthetic polymer. Okay, no, it's a synthetic polymer, and it's hydrophilic in nature, so that it can interact with the tear film. So few of these commercial product uses this PMMA. Here more than one is correct. One correct. Give it a guess. It's fine. So okay, fine. I think you may not have seen the lectures, or seen the lectures you cannot recall. It's fine, totally fine. Uh, these two products, BK Pro and OOKP. So these are the commercial products that predominantly uses PMMA as its core material. So this is the slide in the lectures that discusses the commercial products available to replace cornea. Apart from these two that uses PMMA, there are other products as well, like Miro Cornea. It uses acrylic polymer. Then there is Mi Mekoff, I think Mekoff. So that uses PMMA. There is another polymer widely used for corneal tissue engineering that is called polyhema, right? So this is slightly more hydrophilic. Okay, polyhema is more hydrophilic than PMMA. So that is used as a core material in this particular commercial product, alpha core. So there's like when you get time, slightly go through these slides where they are discussing commercial products for the different tissue engineering applications like bone, vascular tissue engineering, and cornea. It may be asked like in the question. Otherwise, not much relevance to this anyway. Okay, then I'm finished with the questions, but I'm free now if you have any queries from any week content and we can discuss those queries, and I think it will then help you to perform better in the exams. So if any queries, let me know. Any queries from your end or any topic you have not understood in the course and you want me to discuss that topic, I think we'll have time for that too. I'll discuss briefly though, but if your query is specific, it will help.
Ma'am, can you explain the remodeling of bone process once?
Can you repeat?
Remodeling
Explain remodeling process,
of bones.
remodeling process of the bones. All right. So maybe let me find a diagram with which I can help. Okay, so let me share my screen. I hope my screen is visible. Yeah.
Yes, ma'am.
This is a very good diagram that I found from Google search, simple Google search. So what it says, like, first, this is the old bone, all right? The matrix is your old bone, which needs to leave, and new bone is going to form. That is remodeling, that is being shown here. The main cell types that will work in resorption will be the osteoclast cells, make sense? And whatever debris that gets formed, that will be phagocytosed by macrophages here. Then for forming new bone, the osteoblast cells will be responsible here, right? So the osteoblast cells, from where they will come? They will come from these mesenchymal stem cells. They differentiate and form the osteoblast cells. Whereas formation of osteoclast cells, they are actually from the hematopoietic stem cells. So one of the daughter cell of hematopoietic stem cells, they fuse with like cell type coming from mesenchymal stem cells, and from that the pre-osteoclast and then osteoclast cells are formed. So these are the origin of the cell types. Now we can actually divide this process into phases. The first phase is activation, like the process has to get activated, and like maybe some stimuli or something like that, that will activate the process, and that activates the cells. that are going to play a role here. Means formation of the osteoclast and osteoblast, so they will get activated. Then comes resorption process. So in resorption, here is the osteoclast cells that are going to play the dominant role here. These are the osteoclast cells. They will start dissolving out, breaking down the bone construct. Then reversal, like, okay, fine, the damage, whatever damage the osteoclast cells has done, that has to be removed, right? That debris has to be removed. So macrophages will come here, and then comes the process where new bone is going to be formed. So these are the osteoblast cells. So during activation also, like, these will be there in the area. So these osteoblast cells are now here, and now they're actively working to secrete collagen and secrete, and with the help of osteocyte cells, they will also secrete the mineral component. And in this way, over a period of time, you'll see that this layer of old bone will start degrading and the new bone will start forming. Right? This is another very good diagram that shows that. Okay, first, here resorption phase is happening. Active osteoclast cells are the cells which are going to do resorption. Then there is reversal, all right, where macrophages will also come here. These are the then comes bone formation phase, then mineralization by the osteocytes, and once it happens, and then the resting phase, like the new bone is formed, mineralized ho gaya, the new bone is formed. Then again, whenever the need is, this process will happen again. Okay, in more detail, like, you can find the lectures I just tried to, summarize it in a brief form.
Yes, ma'am. I understood clearly. Thank you, ma'am.
Any other queries?
But for exam, will be having MCQ question or how, ma'am?
I think MCQs will be there. All right. MCQs, fill in the blanks, these type of match the following, this type of questions will be there. Just like the question types you have solved for assignments.
So it will be a ticking kind like that, MCQs.
Yeah, I think this will be online exam, so you have to click like which of the option is correct. Also, like, it's possible that you will get some questions where you have to write it, like you have to write one word or something like that. So that type of question can also be there. Like the similar type of question pattern that you have solved in assignment. There not going to be any descriptive questions. NPTEL doesn't like it. It will give. They will appropriately give you time to. So they don't give you questions like that in assignment. So whatever question type is there in assignment, expect those question types only, but in online mode. So you are used to online mode. So yeah.
Ma'am, ma'am, is it mark for 100 or how much, ma'am?
Marks, I cannot say for sure. All right, I cannot.
Okay,
Maybe ask the instructor via mail. I don't know.
okay. Ma'am, and I have a doubt also. How to calculate the hydrogen bond, ma'am? Like how much is the donor and the acceptor? This calculation I didn't understand from the lecture.
Hydrogen. Can you help me recall from which week it was? I also forgot
One
that.
second.
Yeah,
Oh, it's a week 3.
week three. Which topic? Hydrogen donor and acceptor.
Yeah. The question is like which of the following has one hydrogen has one donor and two acceptor for hydrogen bonding. Options are adenine, guanine, cytosine, thymine. So how to
Okay.
calculate this one?
Okay, okay. So it is talking about um yeah, I think it was in the DNA structure, right?
Yes, ma'am.
They were talking about that self-assembly topic. Yeah. Yeah. I got it from it was coming. Okay. The question that you read from, it's from assignment. Oh, okay. Which of the following? Assignment 3 only. Yeah. Okay. Which of the following has one donor and two acceptors for hydrogen bonding? So you have to go and look. So there is no calculation, first of all. It is not calculated. All right. The explanation that was given in the lectures is based on the structure of adenine, guanine, thymine, and cytosine. You will see that how many hydrogens, free hydrogens are there. So those free hydrogens in the structure will act as uh, donors. So they're free. Whereas the oxygen moieties, which are free, like in form of C double bond O, those regions will be hydrogen acceptors. So depending on how many of them are there, the hydrogen bonds are formed between the bases, adenine, guanine, and that is how the DNA gets stable. That is what she was trying to say during the lecture. So go back, see the structure of adenine, guanine. There is no calculation. Let me tell you, there is no calculation. You have to see the structure of the adenine, guanine, and see the like how the hydrogen and oxygen are arranged around it. So yeah, all right. It's very logical. It's not calculative. It's not quantitative. All right.
Okay, ma'am, I got it.
Try it. You can do it. It's not that difficult. Okay. Yeah. Any other queries? Okay. If not, let me stop the recording. All right.